Can everybody hear me? All right. Tend to be loud, so. Thanks for showing up at 8:30 in the morning. And so I'm going to be talking about work I've been doing lately on modeling quantitative traits adapting to changing environments. And before I dive in, I do just want to make a bit of a plug that we're looking for students and postdocs for the lab on various projects, including modeling projects like what we'll be talking about today. But also if you have interest in population genomics and model systems, we've got some really fun stuff going on with whole genome assembly of population samples of flies, and we have RNA-seq data and things from the same strains. There's a lot of fun stuff to do there, so feel free to drop me an email if you're interested in any of those topics. But regarding what I want to talk about today, I want to start and talk about in general what we want to do in population genetics, which typically involves studying adaptive processes through the signatures that adaptive evolution leaves on patterns of genetic variation. And most of our intuition for the effect of adaptive evolution on levels of standing variation in natural populations comes from these two models shown here. So for a fairly standard fitness model, right, where these are the genotype frequencies and the fitnesses are just like 1 plus SH and 1 plus S. We've got these models of the so-called hard sweep, and we've got the sort of classic work by Norm Kaplan and colleagues. And this is a model of a mutation that's unconditionally beneficial when it first arises in the population. And if it's lucky enough to fix and it's strongly selected, it'll do so very, very quickly, and it'll reach fixation. It'll reach a frequency of 1. And the other model, which has been getting a lot of discussion in the last decade, is a so-called soft sweep model, which is perhaps a bit of an unfortunate term, but it is a model where you've got standing variation. Maybe it was deleterious, maybe it's neutral, and then at some point it shifts to become adaptive. And so in this figure from a paper about 10 years ago by Molly Jaworski and colleagues, you've got a mutation that's drifting around here, it's neutral, then at some point it becomes beneficial and it rockets through the population. And the main difference between these two models in terms of their effect on linked genetic variation is the number of haplotypes that they'll drag up with them. A sweep from standing variation that becomes selected at a higher frequency will tend to be associated with more haplotypes and have a less pronounced effect on reducing variation. And so the patterns predicted by these two models are pretty well known, but what I want to talk about a bit is their limitations. So they're both models that predict the beneficial mutation fixes, all right? And so that makes a fairly strong, and they fix quickly. So it makes a fairly strong prediction that if there's been a selective sweep in a region, there won't be any genetic variation relevant to that phenotype at that locus. And when we imagine recurrent beneficial substitutions in populations, the population will just, under a recurrent hitchhiking model, as Kaplan and all considered, you'll just keep sweeping these mutations at the same effect size over and over and over again at, on average, the same time between sweeps. And there's no notion of the population the population phenotype adapting. Really, the phenotype is just to maximize fitness at a particular locus. And there's a bit of a disconnect between these models and what we see in natural populations in terms of adaptive genetic variation. And these are two examples chosen from two things that I really like, which are Drosophila and copy number variation. The figure on the left is the genetic basis, the major effect locus involved in DDT resistance in Drosophila, and it involves a duplication of CYP6G1, which is a cytochrome P450, and over the years, this locus has evolved with more and more duplicates and rearrangements and transposable element insertions, and DDT resistance isn't fixed in any population, okay? So you've got recurrent modification, but no fixation of a trait that has clear adaptive value in areas where DDT is used. On the right, we have a similar story. This involves a duplicate, well, involves the gene Chekhov-1. Variation in expression level of Chekhov-1 is associated with sigma virus resistance as well as insecticide resistance. The major allele that confers resistance involves a dock element insertion that upregulates the locus. And then there's a very, very rare copy number variant in nature that is that duplicates the gene and is even more resistant. And these are just two examples of adaptive traits, and there's presumably fairly strong selection on these phenotypes like insecticide resistance, but the causal mutations aren't fixed in the population. And so what I want to talk about today is models where, and these are old models, okay, so we're sort of going back in time, half a century or so here. There are a lot of models where populations, the phenotype adapts quickly, but fixation of the relevant mutations is just not the outcome, all right? And so these are going to be models where basically quadratic selection on trait values. And so instead of a selection coefficient, we have some effect size on the trait A, all right? So the heterozygote will have a trait value of A over 2, and the homozygote a trait value of A. P0 is some optimum trait value, and for a single locus with Hardy-Weinberg genotype frequencies, the mean trait value will be the frequency of little a, the little a allele, which is Q, times the effect size of the mutation. And what's different about this model from the classic models is how fitness is treated. Fitness is given by this function here, so it depends on the squared deviation from the optimum, and VS is the total variance in the intensity of selection, and it determines like how strongly deviations from the mean trait value or from the total deviations from the optimum are selected against, all right? And this model was first really discussed by, as far as I know, by Robertson about 60 years ago. And what we're going to do is just some really simple extensions to this model where he considered the optimum trait value as the current population mean and considered recurrent selection on the mean. And we're just going to say, well, what happens when the optimum changes and it's away from the mean, and just look at what happens to change of allele frequency. So it's very simple extensions of that work. And so we can write the change in allele frequency when P naught, the optimum trait value, is not the mean. We can write it in terms of allele frequencies, but it actually, I think, is a little more helpful to write it in terms of the mean trait value of the population at that time. And we see that the change in allele frequency depends on where the population is on average in terms of its average trait value with respect to the optimum, and also the effect size of the mutation plays a role here. And just staring at these equations for a second shows you that the change in allele frequency can be positive or negative depending on where the population is with respect to the optimum and how large an effect the mutation has. And so this is the same as in Robertson, where the selection on the single locus contributing to this trait is frequency dependent, all right, in terms of the direction of allele frequency change. And also for these models, the equilibrium allele frequency for a single site is easy to work out, and the equilibrium allele frequency will not involve fixation if the effect size with respect to the optimum falls within a certain range. So in other words, there are just conditions where the population, the population will adapt to the new optimum, but the mutations won't be fixed, all right? And so what we're going to do is consider the effect of considering this model on genetic hitchhiking. So, and what we're going to do here is turn to simulations in order to study a multi-site model here under a pure additive effects model. And the reason for doing this goes back to a key insight from Robertson, which is if there are 2 sites contributing variation in the trait, or 3 sites, they all can't simultaneously be at their equilibrium. Because if site A is at its equilibrium and site B moves to its equilibrium, the mean trait value moves, and then the population is no longer at its equilibrium, and there will be selection. And so the population will start adjusting the allele frequencies of all of these loci in order to minimize the variation in fitness in the population. And what we're going to do is use simulations to look at those dynamics. And these are going to be forward time simulations, and the setup of the simulations is quite simple. We have a single contiguous genomic region with a recombination rate of 50 centimorgans. Deleterious mutations occur at this rate, μd, per gamete per generation, and the effect sizes are Gaussian with a mean 0 and a particular standard deviation. Neutral mutations occur at rate theta uniformly across the region, so in sort of to a loose first approximation, this level of variation is like 100 kV in humans, all right? But it's just to let us throw down neutral mutations so we can see the effect of selection on the variation at these neutral sites. And so if the total variation in selection intensity is 1, I'm going to show you results for this deleterious mutation rate. I've done more, but our cluster went down for maintenance this week, so the figures didn't happen. And I'm going to show you results for a fairly large variance in mutational effect sizes. And with assumptions like this, under a fairly wide range of conditions, including gamma distributions of effect sizes and things, for a population evolving to an optimum trait value of 0, the average genetic variance at equilibrium under this mutation selection balance is given by this formula. So it would just be 4 times the mutation rate to mutations affecting the trait value. And importantly here, I'm going very, very simple, all right? Pure additive effects model, no pleiotropy, all of those things are important, but I think it helps to start from the simplest model possible. Everything's going to be shown for population size of 1,000 based on 1024 replicates, and the software here is based on some C++ code that I published a couple years ago, which is made more user-friendly through a Python package that we're developing. It's open source, it's available, you can get it now, and it's designed to run on clusters in a parallel fashion. And so I show this plot to sort of orient you with regards to what I'm going to be talking about in all of these plots, which is the optimum shift occurs at time 0. The population's been evolved for 10n generations. And then the optimum in this figure shifts to 0.5. And here's the mean trait value. I've just multiplied it by 2 to sort of separate these lines out. So the mean trait value, we get rapid phenotypic adaptation, and on average, the population hits the new phenotypic optimum in about 0.25 n generations. And variance in fitness and genetic variation increase after the optimum shift, and then slowly start to decrease back towards equilibrium after the populations hit the new optimum. But importantly, on average, when the population hits the new optimum, the mean fitness shown here in purple hasn't recovered. And that's because the populations hit the new optimum, but there's still segregation mode in the population. And so there's variance in fitness. And in the interest of time, I'm just going to move on and show some of the interesting dynamics here. So here we have the average effect size of mutations at fix during these simulations as a function of time since the optimum shift. And this is where we're seeing differences. The model's inherently different from the standard model. The effect sizes are not constant. Mutations that occur before the optimum shift, those are by definition soft sweeps that then fix after the optimum shift. They have sort of modest effect sizes. Mutations that arise shortly after the optimum shift, they're hard sweeps by definition, they're new mutations. They have large effect sizes. By about this point in time, the population's adapted, and we have smaller and smaller effect sizes fixing over time. But during adaptation, because of this idea where all the mutations can't be at their equilibrium simultaneously, we get these really sort of interesting dynamics. So here are the frequency trajectories of all the mutations that have arisen during a simulation. And the point here is that because there's this variance in load, segregation load, which imposes variance in fitness, we have mutations affecting fitness repeatedly evading the population until all high-frequency mutations fix, okay? And so this is quite a bit different from the standard model, and the interesting implication is that at any time point until this process is resolved, we'll see mutations that would be interpreted as balanced polymorphisms, but they're not the evolutionarily stable outcome. And this has a lot of implications for how we think about observations in human and fly population genetics, as well as experimental evolution. And finally, just the patterns of hitchhiking here can be fairly short-lived. Haplotype-based statistics like IHS seem to do very well at detecting hitchhiking, but when the optimum shift is fairly modest, things like Tajima's D don't have much of an effect. And so I'll thank you for your time, and move on to the next speaker.